So it's about 19 hours and the class starts now. Like I promised that we are going to have a class that is around 19 hours. I guess it's beyond 19 hours, but this is a good start. This is a good time to start. So now, like I said, we are going to start first by looking at the microscope. So microscope is the first part that we are going to look at. And now since we are going to study cells after this, you should understand that cells are tiny in such that they cannot be seen with our naked eyes. So we need the aid of the microscope. So the microscope is simply the instrument that magnifies the small objects that we cannot see with our own eyes or our naked eyes, in this case cells. So it's very important, or let me say the microscope is an important instrument when it comes to a study of cells. So please, I can see people joining. You can just confirm to your end that you're able to get me clearly, and of course so that we can proceed. Are you able to get me clearly to your end? I'm waiting to hear from you. Are you able to get me clearly? Can we start? Okay, so we start on the microscope. What is important is to look at the parts and the functions. What are the functions of the parts that we have? So we're going to start with the eyepiece. What is the function of the eyepiece? What is the function of the eyepiece? As you can see here, the eyepiece is right here, okay? So now this is where you put your eye when you want to observe something in a microscope. This is where you put your eye. So the eyepiece, we can say it contains magnifying lenses, and it is where we put our eyes in order to view the object that is the one that you are studying or under the study. So the eyepiece simply contains the magnifying lenses, and that is where we look through. Then the other part that we can look at here, we can move to the body tube. What is the function of the body tube? So now, as you can see, on top of the body tube, that is where you find the eyepiece, and down here you find the coarse or the revolving nose here, the one that has some objective lens here. So the body tube, we can say it holds the eyepiece and lenses in position. So hold the eyepiece and the lenses in position. That is the function of the body tube. Then we can move to, let us go to this part. So for adjustment knobs, we have the big one and the smaller one. And the big one is called the coarse adjusting what? Knob. Sometimes they are found attached on the same part. You have the smaller one inside and the big one outside. So that is how sometimes they are on the microscope. But in this case, they are separate. So as you can see, the coarse adjusting knob, what is the function? So the function of the coarse adjusting knob in this case, that is the coarse adjusting knob. So for the coarse adjusting knob, the function is to move the body tube to produce a large changes in focus. Okay, so for this one, when you turn this one, you are moving the body tube. In short, you are moving the body tube at a long distance. Okay, in short, you are, let me say, or it moves the body tube to produce the large changes in focus. I think that is accurate. Then we have the fine. Now, the opposite is true. For the coarse, we said it moves the body tube to produce large focus. This one, it moves the body tube slowly, okay, to produce a small focus or changes. So it moves the body tube to produce a small changes in focus. So this one is the one that you use now. If you want to make the image or the object clear, okay, to make the image of the object clear. So you move or use the fine adjusting knob. as you move it slow, it is able to move slowly as it magnifies the object. So that is very important. Now, what about the revolving nose? What about the revolving nose? So for this one, it is a circular metallic piece holding the magnifying lenses to the tube. So this one, we can simply say, in short, it holds that is it holds the magnifying lenses. Okay, as you can see here, they are objective what? Lenses that you find here. They are of different magnifying power. Okay, we have lower magnifying power, and of course we have higher magnifying power here. So they are normally labeled times 40, so it means this one is able to magnify 40 times, times 10, and times 100. Okay, so that is very important. Then, of course, we have here the stage. Okay, the stage should be indicated here. This is the stage. And on the stage, okay, let us look at the stage first. So for the stage, that is where you put, it's a flat surface on which the slides are placed. Okay, that is where you put the slides. Now, slides, there's what you call this thing. What do you call this? That is where you put the slides. The name has even gone. Okay, so where you put the slide, that is where now you prepare a slide, then you put it on, I wouldn't say that is a stage creep. The stage creep is something different that holds the slide. Okay, so here on the stage, that is it's just a flat surface where you put what? Where you put your slide. Then there's a slide clip that holds the slide from moving. Okay, so that is very important. Then, of course, we have this condenser here. What about the condenser? So I forgot to mention that these microscope, they come in different types. We have what we call electric microscope, the one that uses electricity and it has a light at the bottom here. Okay. But so for electric, it has a light. Okay. That switches that you can switch on as you use the microscope. But also we have the one that uses the natural light, which is from the sun. So depending with what is here. So if it's a light or mirror, it reflects light toward the stage, and this passes through the condenser. Then, of course, if it's a light also, that is the bulb that is here, that is powers, that the source of light here also, the function is the same. That is just to provide the light so that you can clearly view the image on the stage clearly. And that amount of light that reaches the stage is controlled by the condenser. So the condenser, we can say, regulate the amount of light that reaches the what? The stage right here. Okay. Now, if it was a mirror, you can say reflect the light, okay, toward the what? The stage. Okay. And we have the base. The base, that is just where the microscope lies here. Okay. That is the base. Okay, that is the base right there. So just looked at some functions of the microscope. I guess you've had time to look at also some of the functions. So please, those are the functions that we can talk of at a moment for the microscope. Okay, now that we've looked at the functions of the microscope, I think it's the best time now to start looking at cell structure, because we said a cell is just it's a basic unit of life in short, and it's minute in such a way that you can't see it with an aided eye or free eye. Okay, in this case, you require the use of the microscope, and therefore the microscope, we said, is just an instrument that is used to magnify objects that cannot be seen with our naked eyes. Okay, so now let us look at the cell structure now. So under the study or under this part, we are mainly going to focus. We are mainly going to focus on the first part. We are going to focus on that is a simple structure. And I should mention that the cells that we are going to study, two types of cells, that is plant cell and animal cell. Okay, those are the cells that we are going to study, plant cell and animal cell. So the first part, we are going to look at the simplified part of that is plant and animal cell. And that is what you're going to look at. So here are the structures of the simple plant and animal cell. And what is important here is to label the parts. And I guess you are able to see the diagrams clearly. Okay? So this one that you are able to see here is a plant cell. And this on the other side is an animal cell. Okay, so what are the parts that they have in common? Let us start with those that they have in common. What are the parts that they have in common? This is very important when it comes to differentiating plant and animal cell. These are the questions that you expect in an exam. So right here, exam questions or possible exam questions, we can start handling them. So with the animal cell, the outer part is called the cell membrane. Then the inner part here, this part here sometimes is shaded like this. This is what we call the cytoplasm. So the inner part here, it is called the cytoplasm. Then the inside part, the dark part, this one, it is called what? The nucleus. Now, so these are the parts that you find in animal cells. So cell membrane, the outer part, the inside part is called the cytoplasm. Then this dark part is called the nucleus. Now look at the shape. In terms of shape, as we are going to look at the differences, in terms of shape, I don't want you to miss when you find the animal cell. In terms of the shape, you see it's irregular in shape, oval shaped, not really that. So we can just say it is irregular in shape. But look at this. This one is almost a rectangle-like. Okay, rectangle-like. So now what are the parts of the plant cell? So the inner part here, this line that you can see, the dark part, the inside part here, this is the cell membrane. Okay, that is the cell membrane. Then we have the inside part here. This part is called the cytoplasm. And this dark part here is called the nucleus. So I think these three parts are the ones that they have in common in terms of plant and animal cell. But now there are other parts that just you find, or the ones that you find in plant cell, such as this green part labeled here. These are called what? Chloroplasts. Okay, these are called chloroplasts. Then you have this large structure here, this inner structure here. This is what we call the cytoplasm, I mean the vacuole. So please don't confuse the vacuole and the nucleus. The nucleus is normally shaded, and the vacuole, as you can see, is larger than the nucleus and normally centrally located. So this is what we call the vacuole. Then now you have also this outer part now. This outer part. This outer part is all called the what? The cell wall. Okay, this outer part is called the cell wall. But if the pointer is going all the way up to the spaces, look at the lines. If it is going up to the spaces here inside, this is also the cell wall. But when it goes beyond this first part into the inside part, that is the cell membrane. So I've just drawn the lines that separate the two. So the cell membrane is this inside, but outside is the cell wall. But I said within this space here, we can also put cell wall. I guess you've gotten that point. Okay, so now, having looked at the parts of the simplified plants and animal cell, let us look at the similarities between the plants and animal cell. What are the similarities? Now, when you look at the similarities, we look at the parts that they have in common. So in this case, we are going to start with a simple structure. So for the simple structure, the similarities that are there, that they both have what? Cell membrane. They both have cytoplasm. They both have nucleus. Okay, so these are the similarities that are there between that is plant and animal cell. For the simple one, so for the simple one, we can go for these similarities. We are looking at what are the parts that they have in common. So that is what we are looking at in terms of similarities. So take note of that. So for the simplified one, the parts that they have in common, we just have the cell membrane, the cytoplasm, the nucleus. Then now, in terms of, if you want to remember easily, you can come up more like a Venn diagram where you have now what intersect is what they have in common. What does not intersect here In short, what is in the middle here is what they have in common if we are to draw or to put up sets for plants and animal cell in terms of parts. Then what is outside there is what they do not share, or what they do not have in common. So what they have in common is the nucleus, the cell membrane, cytoplasm. These parts are from the simplified one. But when you go to the detailed one, you find mitochondria, ER, which is endoplasmic reticulum, and also the Golgi bodies or Golgi apparatus. So now, this is what we can look at in terms of similarities. Do we have a problem up to this point? Those that are in class, I can see people in class. Those that are in class, can you just confirm that you are able to get me clearly and we can proceed with the class? Are you able to get me clearly and can we proceed? And make sure that you invite others. You know your friends that are writing exams. It might be they are preparing for GCE or they are preparing for that is internal, which is coming in November if you're writing ECZ exams. So just invite, share the link with others, invite them to be part of this live session. We normally have it 19 hours, different subjects. So you can tell them normally 19 hours we have a class, different subject, that is mathematics, physics, biology, chemistry. Okay, and that is biology, physics, chemistry, and mathematics, about 4 subjects. So share with them. If you want the timetable, I can still send to you. Okay, so please let us advance and look at the differences now. So aside from what they have in common here, the differences are these now. The plant cell has cell wall, which means this one does not have a cell wall. Then the chloroplast, the plant cell has chloroplast, which means the animal cell does not have chloroplast. The plant cell has large vacuoles that are centrally located. In animal cells, vacuoles are small, and as cell grows, they tend to disappear. So sometimes you can just say they do not have vacuoles. Okay, so that is those are the differences that you expect to find. So if you are to put them in writing, you're going to say, let me just show you how you present in writing. If they ask you distinguish between or give the differences between plants and animal cell, you're going to say plant cell have cell wall, whereas animal cell do not have cell wall. So please, as you write, you need to make sure that you put the word that shows that indeed you are writing science. Okay? All right. So now, I guess the next one you can formulate the difference. Plant cell have chloroplast, so plant cell have chloroplast, whereas animal cell do not have chloroplast. Then we can talk of plant cells have vacuoles, whereas animal cells, if at all they are there, they have small vacuole. I think those are the differences. So we can proceed now to look at the detailed structure. So for the detailed structure of cells, plants and animal cells, as you can see, the detailed structure, we are looking at the one that is magnified now under electric microscope. Okay? Under light microscope, we're just able to see the simplified part, but when you view it under the power of microscope, you're going to see even the structure that you are not able to see under the simplified one. So now, this is an animal cell, the same one that we've looked at, the simplified one, but you can see there are addition parts that you're able to see. And this is the plant cell, the same one that we've looked at, but here, as you can see, there are some of the structure that you are able to see. Okay, so let us start by looking at the parts that we've looked at, that we are familiar with. So for animal cell, we know the cell membrane is the outer part. Then we know the vacuole. Okay, the vacuole in animal cell, we say they are small. So here you are able to see. Then we know the nucleus. So the nucleus is this part here. Okay, I think these are the parts that we've looked at. And the cytoplasm, please. The cytoplasm. So these 4 parts that I've just labeled are the ones that we've looked at under the simplified one. Then for plant cell, we can also follow it up, the nucleus, We've looked at which other one? Cell wall. We've looked at the cell membrane. The other one is a vacuole. Where is it? This one. So whatever remains, we've not looked at them, which means these are the new parts that we are going to discuss now. Okay? So now, what are the parts that we are coming across for a first time? So the parts that we are coming across for a first time, we have the Golgi apparatus, the centrosome, the mitochondria, the smooth endoplasmic reticulum, rough endoplasmic reticulum, ribosomes or rhizosome. We are coming across the nucleolus for the first time, the nuclear membrane, and of course the lysosome. Then now for this one, also the nucleolus, ribosomes, rough endoplasmic reticulum, smooth endoplasmic reticulum, the Golgi apparatus, the perixome, chloroplast. Oh, chloroplast was there even from the simplified one. So we should have put this in red. So chloroplast was there. But these other parts were not what? Were not there. Okay, so which means these that I've labeled in green are the parts that you are coming across for a first time. And these are the ones also we are going to look at. Okay, so these are the detailed part of plants and animal cell. Now, at this point, what is important? Which part do you consider when it comes to exams? So please, the detailed part, you should know the parts, okay? The parts, how they look. Are you able to tell if you find it in an exam? And also, most importantly, you should be able to give the functions, okay? You should be able to give the functions. So what is next, or something that we are going to look at next, is now to look at their functions. We'll be coming back to refer to the parts and look at their structures, because in looking at their functions, we are going to look at a bit of their description. So that is very important. Okay, so let us go on and look at the functions of these parts. What are the functions of these parts? And we are not going to go into looking at all the parts, but selected parts. And these are the parts that we are going to look at in terms of functions. Okay, so functions of cell parts. So we're going to start with a simple one. So now, here you need to pay particular attention. This is where a lot miss it. The questions are simple, but the way you answer them, the way you phrase your answer is what becomes challenging. So please make sure that you follow me here as I explain what you are required to write to avoid writing paragraphs that are irrelevant in terms of. So here specifically, we are going to give the what? The functions. So now the first one is the nucleus. Okay, what is the function of the nucleus? We all know how the nucleus looks like, where we are coming from. Okay, so the nucleus is the whole of this dark part here, but this is detailed. And equally here, you can find this nucleus. Now, what is the function of the nucleus? So the nucleus controls all cell activities. So nucleus controls all cell activities and is responsible for storing genetic information. Okay, soon you are going to look at genetics, and I think this is a topic that we are going to learn along, so that those that are doing internal in November, you are advantaged as well. At the same time, those that are writing next year. So cells control cell activity and is responsible for storing genetic information. That is the DNA. Then now let us go to the cell membrane. The cell membrane is the outer part of the cell, the animal cell, and also it is found in what? In a plant cell. Now, what is the function? So the cell membrane controls the material that enters and leaves the cell. That is very important. It controls the material that enters and leaves the cell. How does it do it? It is selective repairable. So now listen, where you are going to make a mistake is where you are asked to give the function of the cell membrane, then you say the cell membrane is selective repairable. No, that is just a feature. That is just the adaptation of the cell membrane. The cell membrane, the function is not its being selective repairable. The function is it controls the materials that enters and leaves the cell, and hence it is what? selectively permeable, or the way it adapts to its function is by being selectively permeable, which means it chooses the material to go in and to come out of the cell. And that is all based on the size of the materials. Small material or materials that are small in size, they are able to go in and come out freely, such as oxygen, carbon dioxide, and all those. But large materials are not able to enter, and some are not able to leave the cell. I guess you get the correct function here. Okay, let us proceed to look at now the cytoplasm. What is the function of the cytoplasm? Now, from the simplified one, the cytoplasm, this is a large part here, the large portion of the cell, this inside, where all these parts are found, where the cell, where the nucleus is found, the chloroplast is found, this vacuole is found. In short, all other cells are found. All other parts of the cells are found in the cytoplasm. So now, what is the function of the cytoplasm then? So this is where cell activity takes place. Remember, the nucleus controls the cell activity, and the cell activity are occurring in the cytoplasm. So this is where the cell activity takes place. And now if you are to look at the way it is designed, it is jelly-like, okay? It is jelly-like. So this is where the cell activity takes place, and that is the function. Now, we can also look at the terms such as the protoplasm here. Now, in the case where you find a question, let's say, give the parts that makes up the protoplasm. So the protoplasm makes up, or by the way, or before we even go to what the protoplasm makes of, the protoplasm is made up of the living part of the cells. So if you are to give maybe the parts of the protoplasm, you are just going to look at the living part of the cells. And what are those? The cell membrane, the nucleus, and the cytoplasm. These three parts are the ones that makes up what we call the protoplasm, the living part of the cell. I guess you get that point. Then now let us come to the mitochondria. So these parts that we've looked at, as you can see, they are all found in the simplified one. But what about the mitochondria? So we're going to go to the detailed one. In animal cell, the mitochondria is this that you are seeing here. Okay, let me just use a different color. And of course, we are going to describe it a bit so that you'll be able to relate. Where is the mitochondria? So the mitochondria is this one. Also here we can see the other one. Also here you can see the other one. In other words, it's represented like a rod-like structure. Okay, a rod-like structure. Others will say sausage-like structure. Now, what is the function of the mitochondria? So this is the site, or it is a site responsible for respiration. It is a site for respiration. Now, if it is the site for respiration, then that is where energy is produced. Okay? You know that respiration at the end, it produces what? Produces energy. So it is a site, in terms of function, you can say it is a site for respiration. Okay, responsible for, or that is where respiration takes place. In other words, you're going to find other books describing it as the powerhouse. Powerhouse. That is where energy is generated. So all those parts, all those functions that I've given, you can put any of those. So you can say that is where energy is generated. It's a site of respiration. That is where respiration takes place. All those are the what? The functions that you can talk of in terms of the mitochondria. What about the liposomes? Let us go a bit back and look at the liposomes. So the liposomes are these small or circular vessels that you are able to see. These are what we call liposomes. They are freely found suspended in the cytoplasm, or they can be attached to the rough endoplasmic reticulum. Okay, they are freely found suspended in the cytoplasm, or they can be attached to this part. As you can see, this part, it has some grain-like on it body. Okay, so they can be found attached to what we call the rough endoplasmic reticulum. So the endoplasmic reticulum that has the ribosomes is called rough endoplasmic reticulum because it has ribosomes on its body. The one that does not have is called a smooth. So that is what differentiates the smooth and the rough endoplasmic reticulum. But anyway, we are not looking at the endoplasmic reticulum in terms of function. We are looking at the ribosomes. We are coming back to the endoplasmic reticulum. So I've pointed much at the animal cell. Equally, the plant cell, you do able to find just that this part is not that magnified, but I'm sure you're able to see something like these dots to this structure. Those are what you call ribosomes. Okay, and you can see these, they do not have. Okay, what are the functions of ribosomes? So for the ribosomes, in terms of functions, we are saying it is the site for protein synthesis. So that is where proteins are synthesized. Now, what kind of protein? So proteins, you can find proteins like enzymes and other types of proteins. That is where they are synthesized. So that is where proteins are made in the ribosomes. So take note of this function. So ribosomes, protein synthesis. What about the endoplasmic reticulum? So when you talk of the endoplasmic reticulum, generally, we are looking at, in terms of functions, we can say transportation of substances within the materials. Okay, I mean within the cell. Endoplasmic reticulum, in general, that encompasses rough and smooth transportation of substances within the cells. Now, let us look at each of the endoplasmic reticulum individually. So we are going to start by looking at the smooth endoplasmic reticulum. Okay. Now, I guess you understand or I've told you the difference between the two. So this is the rough endoplasmic reticulum. I hope you're able to see. This is the rough endoplasmic reticulum, and this is the smooth endoplasmic reticulum. No, not this one, please. This one. So this is the smooth endoplasmic reticulum. So the difference is that the smooth endoplasmic reticulum does not have ribosomes on its body, but for the rough one has endoplasmic reticulum. Now, since we are saying the endoplasmic reticulum, the main function is the transportation of substances within the cells. Let us look at what each endoplasmic reticulum transport. So for smooth endoplasmic reticulum, transport lipids within the cells. I guess you've gotten that. The general function of endoplasmic reticulum is transporting what? Transporting materials within or substances within the cell. But specifically, the endoplasmic reticulum, in terms of the smooth endoplasmic reticulum, transports lipids within the cell. Then when you talk of the rough endoplasmic reticulum, transport proteins, that is within the cells. So rough ER transport proteins, whereas smooth ER transport what? Transport lipids. This is very important. These are the questions that you expect in an exam. Then now we have other parts which are called Golgi apparatus. So Golgi apparatus, we are just looking at, in short, Golgi apparatus is just one here, but when you have a lot of these, is called Golgi bodies. Okay, so the power of these is called Golgi bodies, but one is called Golgi apparatus. So what we are looking at in terms of functions here, even here you can see them. These are Golgi apparatus. So a lot of them are called Golgi bodies. This is the mitochondria. Okay, I hope you're able to see that. What is the function of the Golgi bodies? So carry proteins. So the proteins that are made in the ribosomes are carried by the what? The Golgi bodies. So proteins that are carried, examples, enzymes, from where they are synthesized to the site of what? Or site of reaction. So when the proteins, that is enzymes, are made in the ribosomes, they are carried to the part where they perform now their function, or where they are needed for the functions. That is very important. Then the other part that we can look at are the chloroplast. So chloroplast, these are the green part here. Even in detailed one or simplified one, you can see they are only found in plant cell, I mean. So these green ones, okay? So these green ones, this is where photosynthesis take place. So the main function here, you can just say carry out what? Carry out photosynthesis. So that is the function of the chloroplasts. Okay. Then the vacuole. What is the function of the vacuole? The vacuole are only found in plant cell, and the main function they play a role in import. I mean, they play a role in movement of water, okay, in and out of the cell. So this we are going to look at as we start looking at cell specialization. But at this particular point, we can say the function of the vacuole is to regulate the movement of water in and out of the cell, depending with the concentration of the solution in the vacuum. I think we are going to look at the solutions as we go on soon, and this one will get more clear. Then we have the cell wall, which is only found in plant cell as well. Now, the cell wall, to start with, you need to know that it is made up of the hard material called cellulose. So this is made up of hard materials called cellulose. Okay, now what is the function of the cell wall? So one, it protects the cell against mechanical what? Mechanical damage. So one function, it protects the cell against mechanical damage, then prevents the cell from bursting when it has gained a lot of water. Okay, so in short, it withstand, it helps the cell to withstand osmotic pressure. So prevent the cell from bursting when it absorbs or it gains a lot of water. And all this we are going to discuss. We are building on the concept, and soon we'll get to some advanced parts of the topic. And that is where now things start to get interesting. So please, this is the lesson for today. And if you've enjoyed, please don't forget to share with others. Don't forget also to attend the next class, as I'll be announcing the next part that we are going to look at. We are going to start looking at cell specialization. So the next time we are going to start looking at cell what? Specialization. So that is where we're going to discuss now the plant and the plants, I mean cells, different plant cells and different animal cells. Okay, so make sure that you don't miss out that part. So please, as always, if you are interested in the online lessons, please get in touch. If you are interested in the online lesson, please get in touch. We'll be waiting to have you in class. So the number is 0976 402563, or you can use 060643254 or 054806045. Thank you very much, and see you in the next one.